We're going to do Chapter 7 now, and we're going to start talking about nervous tissue, the nervous system. The nervous system structurally can be broken down into two major sections: the central nervous system, which includes the brain and the spinal cord. The actual cells that make up the brain and spinal cord are the nerves, the neurons technically. The neuroglia are the support cells of the central nervous system. The peripheral nervous system is all of the nerves that are coming off of the brain and spinal cord. So some of the nerves come off of the brain. We call those cranial nerves. Some of them come off of the spine. We call those spinal nerves. Cells in the digestive organ walls and sensory organs are also included in this. These include sensory and motor neurons, and nerves in general are bundles of nerve fibers. So in other words, when we're referring to a nerve, what we're actually saying is not a neuron, but we're saying a bundle of axons. That's technically what a nerve is. A nerve cell is one individual nerve fiber. So here you see what I mean with the different divisions. The central nervous system being the brain and spinal cord. That's where the majority of your neural tissue is. The peripheral nervous system contains the cranial nerves, the spinal nerves, as well as a group of nerves called the enteric plexus, and our sensory receptors are included in the peripheral nervous system as well. These would be like our thermoreceptors to sense temperature, mechanoreceptors to sense touch. Our peripheral nervous system can further be divided into the afferent system and the efferent system. The afferent system is our sensory nervous system. Think of senses feel good, something smells good, or something sounds good, or something feels good. That A and afferent can make that association for you. So the afferent nervous system is our somatosensory receptors within our muscles, within joints, and within our skin. So in other words, it's communicating to the central nervous system our overall body position and whether or not various muscles are flexed or extended. Our special sense receptors include the receptors within our eye, ear, nose, and tongue for our vision, our hearing, our sense of smell, and our sense of taste. Autonomic sensory receptors are also within our internal organs, and they're going to communicate with the central nervous system things like whether or not your stomach is empty, your stomach is full, or you need to go to the bathroom. The efferent or motor nervous system is composed of the motor nerve fibers. These motor nerve fibers regulate the activities of our skeletal muscles as well as the gland tissues. There's the somatic nervous system, which is our voluntary skeletal muscle activity, the autonomic nervous system, which is our cardiac and smooth muscle, as well as some gland tissue, and then the enteric nervous system, which is the involuntary functions of our digestive tract. This video here goes through the overall structure and function of the nervous system as well as how it is organized. Here you can see another graphic organizer showing you how our nervous system is organized. This one is branching off of our central nervous system. You can see the central nervous system being our brain and spinal cord. This purple box is representing our peripheral nervous system. The peripheral nervous system can be broken down into somatic, which means skeletal muscle, autonomic or automatic, those are the things that you don't need to think about to do, and then the enteric nervous system, which would contain our digestive system. Let's take a look at the afferent, efferent fibers here. So if you remember, all of the input coming into our central nervous system is going to be sensory or afferent, so that's what all of these blue arrows are representing. Notice the enteric sensory receptors are going into the central nervous system. Autonomic sensory receptors going into the central nervous system. Somatic sensory receptors going into the sensory nervous system. Or the central nervous system rather. So if we were to break this down further, let's take a look at somatic nervous system here. You have somatic and special sensory receptors. This would be like sense of touch, sense of temperature, etc. Coming into our central nervous system and saying, hey, it's hot in here, or hey, we have an itch, or something like that. Our central nervous system is then going to say, okay, we need to, scratch our nose, or what have you. So these red lines or pinkish lines are representing the efferent motor pathways. The somatic motor neurons are going to carry that voluntary information to the skeletal muscle. These green boxes are called the peripheral effectors. In other words, these are the guys that are going to actually move or respond to the central nervous system's impulse. If we look at our autonomic nervous system, you have those autonomic sensory receptors communicating with our central nervous system. Various information, for instance, blood pressure, maybe blood pressure is increasing or decreasing. And so our central nervous system is going to send an efferent or motor response through an involuntary pathway. This can either be a sympathetic or parasympathetic effect, and we'll look at what those are in a minute. And that information travels to the smooth muscle, cardiac muscle, or maybe even some glands. In the enteric nervous system, notice the relationship between the autonomic nervous system and the enteric nervous system. The enteric nervous system is kind of a subdivision, for lack of a better term, of the autonomic nervous system. So the enteric nervous system is just a branch from the autonomic nervous system. The sensory receptors from our autonomic or our enteric nervous system rather are going to send information to the central nervous system. For instance, maybe you need to go make a bowel movement, and your central nervous system is going to respond and say, oh, okay, well let's go ahead and contract our large intestine to get that fecal material out of here. So then the enteric motor neurons are going to involuntarily contract, and the smooth muscles, glands, etc. will receive that contraction information. They will contract, and that's when you say, whoa, I need to go use the bathroom. So this is looking at the same information but in word form instead of in graphic form. The sensory input is our peripheral nervous system. Neurons are going to sense the change in environment, ultimately send that information back to the central nervous system. There's an integrative function or a information processing that has to go on, where our central nervous system is looking at the input and then deciding what to do. Is it going to respond, or is it just going to take that information into account with whatever the following decision steps are? This is where conscious decisions, which require perception and higher level processing, takes place. Our motor function is going to be the information output. That's when the sensory neurons, or rather the central nervous system neurons, are going to send commands to muscles or glands to carry out the responses. Think about what happens to you when you see a ball coming towards your face, or what happens in the physician's office when he or she taps your knee with that reflex hammer. That's all sensory input in, a motor function out. So let's talk about a neuron. Let me backtrack just a minute because I'm not sure if this PowerPoint goes into sympathetic versus parasympathetic. I thought that I had a slide in here, but maybe I didn't. So let's revisit this briefly before we go into the structure of a neuron. Autonomic motor neurons, as we said, this is all the automatic nervous system, and I mentioned it could be sympathetic or parasympathetic. Sympathetic is that fight or flight response, so this is that increased heart rate, increased respiration rate, increased awareness, increased metabolism. The parasympathetic division is more the rest and digest. So this is when your muscle activity slows down, your metabolism slows down, your digestive secretions increase, your digestive movements increase. So this is why they say that you shouldn't go swimming after you eat, because after you eat, your parasympathetic division is dominating, and that's that rest and digest. You just ate, so all of that food now needs to be digested, but swimming brings about the sympathetic nervous system. So the hem and haw between the sympathetic and parasympathetic is what could be dangerous for you if you're swimming while you're digesting, or at least so my mom told me. So let's go back to this slide, sorry about that little detour, and talk about an organelle, or rather a neuron. Oh my goodness, it must be late. So a neuron is an electrically excitable cell. So it's a highly specialized cell that's specialized for transmitting electrical impulses. Some of the organelles inside of the neuron include the endoplasmic reticulum, mitochondria, the nuclei. Those are all maintained within a portion of the neuron called the cell body. The neuron itself has nerve fibers coming off of it, or long strands or extensions coming off of it. One long strand is called the axon. This is going to carry the electrical impulses away from the cell body. There's only one axon per neuron. The dendrites are going to conduct the impulses toward the cell body. So these are associated with sensory nerve receptors. They have multiple dendrites per cell. The dendrites are on the receiving end of the electrical impulse, and then they conduct that information toward the cell body. The cell body then transfers that information to the axon, and the axon can transmit that information to either another neuron or possibly a muscle fiber or gland tissue. A ganglion is a collection of cell bodies that are within the peripheral nervous system. A nuclei is the collection of cell bodies in the central nervous system. So when we're talking about nuclei in the brain, we are talking about cell bodies. When we're talking about ganglion in the, say, the peripheral nervous system in the abdominopelvic cavity, for example, we're still talking about a group of cell bodies. It's just the location that's going to differ. So here you see what I mean with the overall anatomy of a neuron. Here's the cell body. You can see the nucleus. There are specialized structures called Nissl bodies in the nucleus. It's endoplasmic reticulum and ribosomes. You also have, coming off of the cell body, lots of branches. Remember, a cell body will only have one axon coming off of it, but can have many dendrites coming into it. So the information is going to come from another cell, possibly right over here, connecting with these dendrites of this cell. That cell is going to conduct its electrical impulse down the dendrite, across the cell body, and then down the axon to the end of the axon, which is called the axon terminal. And then these axon terminals meet up with other dendrites, or maybe some muscles, if this is an efferent fiber, etc. If we look at the some of the special labels here on this cell, you can see that this axon is surrounded by special cells, special neuroglial cells, called Schwann cells. These Schwann cells help to maintain what's called a myelin sheath around the axon. A myelin sheath is going to speed the conduction of action potentials. An action potential, if you recall from our chapter on muscles, is an electrochemical signal that carries down or carries across the cell membrane. So the action potential that's coming from the dendrites is going to cross this cell body, and then it's going to go across what's called the axon hillock, which is the very first segment or beginning part to the axon. And then the action potential is going to jump over these myelin sheaths and hit what's called the nodes of Ranvier, or the nodes. So instead of this action potential having to meander all down every single piece of this cell membrane, that myelin sheath allows that action potential to just jump, 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 until it gets to the very end, the axon terminal, where it can synapse or meet up with another cell. So, neurons are classified by the number of cellular extensions. The neuron we were just looking at is called a multipolar neuron. This is the majority of the neurons within our central nervous system. It has a cell body, the dendrites, where'd my mouse go? Here it is. A cell body, the dendrites, and one long axon coming off of it that can extend onto several axon terminals. A bipolar neuron are really only found in a few special places like the retina of the eye, parts of the inner ear, parts of special parts of the brain, the olfactory areas of the brain. And it's called bipolar because on one side of our cell body we have one main dendrite which branches off, and then on the other side of our cell body we have one axon which then branches off. So it's called bipolar because it has two sides that almost look like they're identical. A unipolar neuron has a fused dendrite and axon formation. So these are, most of our sensory neurons are this type of neuron. So when you are feeling something that's hot and that information is traveling up through your spinal cord to your brain, that's all traveling through these unipolar neurons. And you can imagine why that would be beneficial. The action potential does not have to go through the cell body. It can go through the dendrites and then right down the axon to the axon terminals, as opposed to here where it goes through the dendrites, then through the cell body, then down the axon. So most of our sensory neurons are unipolar because that's going to speed up the conduction of those important sensory impulses. So our sensory and afferent neurons, as I was describing, they have special receptors and they're connected to sensory receptors. We have somatosensory and autonomic sensory neurons. Those are all unipolar, as I was just describing. We have special sense receptors, which are typically attached to bipolar neurons, as I described as well. Our motor and efferent neurons are mostly multipolar, and they carry information from the central nervous system to various effectors such as muscles, through cranial nerves or spinal nerves. Interneurons are mostly multipolar neurons, and they have a very large population. In other words, the majority of our neurons within our central nervous system are interneurons. Interneurons are the relay center between the afferent and efferent neurons. So you can imagine the role that they play in information processing. Afferent sensory neurons bring the information in. Interneurons are then going to decide what to do, and then the efferent neurons will carry out the motor command. So they extend for very short distances. They contact nearby neurons within the brain, within the spinal cord, or within specific ganglia in the body. And if you recall, ganglia is just a group of cell bodies. So this animation here goes through neuron structure as well as function. Let's talk about neuron function. Neurons, as we mentioned, are specially designed cells for conducting action potentials or electrochemical signals over a range of distances. The distance can be of just a few millimeters, or it can be up to a meter long. Think about the length of your leg. Those nerve fibers traveling up and down your leg need to be very efficient, and they're also very long. Myelin, as I mentioned, is that lipid sheet that kind of forms that protective covering to help insulate our axon. It also helps to speed the conduction of those action potential impulses. So, there are neuroglial cells as well as neurons that make up nervous tissue. The neuroglial cells are the special supporting cells. Within the central nervous system, we have different ones than what we would have in the peripheral nervous system. Within the central nervous system, there's microglia. These provide protection against microbial organisms. They're kind of like a mini immune system within our central nervous system. The astrocytes help form a structure called the blood-brain barrier. This is going to help protect neurons from pathogens and other harmful chemicals. This is why not everything you bring into your body will mix with your brain. Your brain is actually protected from your blood. Whatever you eat or bring into your body goes directly to your blood. Very few of those substances can breach that blood-brain barrier. Alcohol and some drugs like cocaine can breach that blood-brain barrier, which is why you get drunk or you get high when you take those drugs. Oligodendrocytes are the types of cells which produce myelin in the central nervous system. Before I identified them as Schwann cells produce myelin in the peripheral nervous system. Ependymal cells are the special cells which secrete cerebrospinal fluid. Here you see some examples of our central nervous system neuroglia. This is a layer called the pia mater. This is one of the meninges that we'll look at later. It's one of our protective boundaries. Underneath the meninges, or deep to the meninges, we have all of our nervous tissue. Here you can see a gap in between where the pia mater is and this layer called ependymal cells. These ependymal cells, as I mentioned, secrete cerebrospinal fluid. These big blue star-shaped cells are called astrocytes. They're there to help create that blood-brain barrier. They also play a role in learning and memory and help with the growth and migration of neurons during embryonic development. These large cells here, which have a very defined cell body as well as dendrites and an axon, these are multipolar neurons of the central nervous system. So here you see 1, 2, 3 of those. Notice this kind of spider-like creature called an oligodendrocyte, which is wrapping its cytoplasmic extensions around the axon of these neuron cells. That's forming that myelin sheath. Over here, this little green guy is a microglial cell. This is acting like a little Pac-Man, eating up any cellular debris that may accumulate. The neuroglia of the peripheral nervous system, as I mentioned, is slightly different. In the peripheral nervous system, there's satellite cells, which are supportive cells within the ganglia, helping to regulate the exchange of fluids and gases between neurons and the interstitial fluid. And then there's the Schwann cells, as I've mentioned before. Schwann cells produce the myelin around a single axon and can aid in speeding the transmission of action potentials. This animation here goes into a brief description of membrane potentials. Let's talk about membrane potentials. Most cells, when they're sitting there not doing anything, maintain what's called a resting membrane potential, or RMP. Relative to the external environment, most cells are negative. So let's think about why a cell would be negative. If you recall from chapter 2, some ions carry a positive charge and some ions carry a negative charge. If the number of positive ions outside the cell is higher than the number of positive ions inside the cell, then the inside of the cell would be more negative than the outside of the cell. So, with that said, the resting membrane potential of a neuron is negative 70 millivolts. That's the measurement of voltage. And the ions that cause this negative 70 distribution are sodium and potassium. Sodium is a relatively small positive ion. Potassium is a slightly larger positive ion. So what we're saying when we say that the cell has a resting membrane potential of negative 70 millivolts is that the number of sodium and potassium ions outside of the cell is greater than what it would be inside the cell. This animation goes through the idea of resting membrane potential. A graded potential is a localized change in membrane potential. So the membrane potential becomes more positive. We say that the cell has depolarized. We use that term when we were talking about muscle contraction. If the membrane potential becomes more negative, we say that the cell is hyperpolarized. This is when stimuli in the environment are going to cause the ion channels to either open or close. A graded potential can cause the cell to slightly depolarize or slightly hyperpolarize, but doesn't always lead to an action potential. If the graded potential allows enough change or enough depolarization with our cell, in other words, our cell gets closer to zero or less negative, then an action potential could occur. We say that that reaches threshold in order for that action potential to occur. Here's an animation about graded potentials. An action potential is when that electrical signal propagates or moves along the membrane of a neuron. The beginning phase is the depolarizing phase. This is when the sodium channels open and sodium rushes into the cell. The repolarizing phase is when the sodium channels close and potassium channels open and potassium rushes out of the cell. This animation goes through action potentials. Ultimately, with an action potential, the greater the stimulus, the more action potentials will occur in a given period of time. The magnitude of depolarization is always going to be the same. So there's no really depolarized state for a cell. Even though it's getting a huge stimulus, it's always going to depolarize even if the stimulus was small. The greater the stimulus, that's going to increase into higher frequency of action potentials, not a larger action potential. All action potentials are created equal, but with a big stimulus, you'll get more action potentials. Here you can see the process in a nutshell. As I mentioned, at a resting state, our cell is at negative 70. That's what you see here on this graph, negative 70. What that's telling you is that there are high numbers of sodium outside the cell and high numbers of potassium inside the cell. When our sodium channels open, because there's a higher concentration of them outside of the cell, our passive transport's going to occur and sodium's going to rush into the cell. This is going to cause our cell to become more positive. Sodium's a positive ion. We take these positive ions from outside, stick them inside. Our cell's going to become less negative or more positive in this case. That's that depolarization, as I mentioned. Once our cell reaches a certain level, around positive 30 millivolts, the sodium channels close, the potassium channels open, And since there was a relatively high concentration of potassium inside the cell and a relatively low concentration of potassium outside of the cell, potassium is going to undergo passive transport and just scoot right on out of the cell. So our cell now is going to lose positive charge and it's going to repolarize. Once our cell reaches that negative 70 millivolts, our potassium channels will then close and our cell is back at its resting state. This animation goes through the propagation of nerve impulses. This animation goes through membrane transport proteins involved in action potentials. This animation goes through factors that affect the conduction rates, including myelination. So let's talk about what's happening at the synapse. The synapse, if you recall, is the gap between a neuron and either another neuron or a muscle or a gland cell. When we were in our muscle chapter, we called it the neuromuscular junction. If you recall, that axon terminal kind of hovered over top of the cell membrane of the muscle cell. That gap is called a synapse. Usually, it's a chemical transmission between our nerve cells. In other words, there is a chemical that's exchanged from one cell to the next cell, which transmits that action potential. Sometimes it's an electrical impulse, like in the case of our cardiocytes, but for the most part it's going to be a chemical, like acetylcholine, that transmits from one cell to the next. Action potentials always travel down to the axon terminals. Remember, the axon is carrying that action potential away from the cell membrane. When it does, calcium channels open, and that inward flux of calcium is going to trigger the outflux of the neurotransmitter. The neurotransmitter is a chemical messenger. In our neuromuscular junction, we were looking at the neurotransmitter acetylcholine. Neurotransmitters will bind to specific receptors on a postsynaptic cell, and that's going to trigger the action potential or opening of sodium channels in that cell. Here you see the basic anatomy of synaptic transmission. So here we have our axon coming away from our cell body, that action potential is traveling down, and these are the little synaptic vesicles which hold the neurotransmitters, maybe acetylcholine, maybe epinephrine, maybe norepinephrine, maybe dopamine, maybe serotonin. There's a number of different neurotransmitters. But as this action potential comes down, our sodium is rushing in, calcium's going to rush in also. As that calcium rushes in, it's going to push these synaptic vesicles out through active exocytosis, and in that process, those neurotransmitters can then bind to that postsynaptic cell, that cell that's on the other side of the synapse, and when those neurotransmitters bind to those receptors, it causes those sodium channels to open, and the sodium rushes into the cell, and then the action potential can propagate and continue in that postsynaptic cell. So, synaptic transmission is when the single neuron produces only one type of neurotransmitter. Generally, synaptic transmission goes only one way from presynaptic to postsynaptic. It doesn't go back up the other way. That's why we have sensory neurons, which carry information in one direction, and motor neurons, which carry information in the other direction. Some of the synapses can be excitatory synapses and cause depolarizations, but not always are action potentials telling the cell to do something. Sometimes action potentials can be inhibitory and telling the cells not to do something, causing a hyperpolarization. Drugs and some toxins, some can enhance the depolarization and some can inhibit the depolarization, causing a hyperpolarization. This is why, for instance, if you take Tylenol or ibuprofen because you have a headache, what that's doing is it's inhibiting the transmission of the sensory impulse that your head is hurting you through those sensory neurons to your central nervous system. So you forget or you're not aware that you have a headache because the information cannot travel through the sensory neurons because they've been inhibited. So this animation here goes through the events of the synapse. Let's talk about neuron communication. As I was mentioning, the idea of those afferent fibers touching a pen is going to stimulate the sensory receptors within the finger. Here you see those sensory receptors in the finger. Those sensory receptors are going to then be triggered, and it's going to cause the sensory neuron or afferent fiber to carry its electrical impulse, its action potential, back to the spinal cord. That spinal cord is then going to relay information up to the brain, to an interneuron. If you remember, the interneuron is the relay center between the sensory and the motor fiber. That's going to communicate with our cerebral cortex, the higher levels of our brain, and say, hey, we're touching a pen, we're writing, this is the word that we're writing, etc., etc. The cerebral cortex will then say, oh, okay, this is how, our muscles need to contract so that we can write the letter A. And so it's going to send that information down this maroon colored motor neuron, leaving our spinal cord, going back down to our muscles so that we can grasp the pen, flex our wrist, and write the letter A, for example. So let's talk about some protective coverings. You may have heard of meningitis before. Meningitis is the swelling or inflammation of or infection of the meninges of the central nervous system. The meninges are connective tissue membranes. There's 3 of them: the dura mater, the arachnoid mater, and the pia mater. The dura mater lies just underneath the bone. So in other words, on the underside of the cranial bones, usually attached to the cranial bone itself, is the dura mater. It's very, very tough, hence the term dura, like Duracell batteries, which are very hardy batteries, so they say. Then you get this purple layer, which is the arachnoid mater. Notice this purple layer is separated from the other layers by these kind of spider web filaments, hence the term arachnoid matter, like a spider web, like arachnophobia, fear of spiders. Then you have this pia mater, which is this red or maroon line. The pia mater is fused with the brain, and the pia mater holds blood vessels in place. The pia mater is the deepest meningeal layer, and usually if you're going to do a brain dissection, the pia mater is still intact on the surface of the brain, almost impossible to separate from the surface of the brain, whereas the arachnoid and dura mater are removed already. The cerebrospinal fluid is a fluid that's derived from blood. It's manufactured within special cavities of the brain called ventricles. It circulates through the brain ventricles, around the brain and spinal cord. It actually circulates in this small space in between the arachnoid matter and pia matter. It's called the subarachnoid space. And the cerebrospinal fluid functions to maintain that blood-brain barrier so the blood doesn't come in contact with our neural tissue. It helps get rid of waste as well as deliver oxygen and glucose to our neural tissue. It also serves as a cushioning for protection, kind of like a big water balloon surrounding our brain. Here you can see those ventricles. Sorry, my computer's going crazy. Here you can see those ventricles within the brain. If you do a brain dissection in lab, you will very clearly see these ventricles. The models in lab will also show these ventricles to you. They're literally spaces and holes within the brain. Our brain is not a solid mass. There's holes within them called ventricles. We have 2 lateral ventricles, a 3rd ventricle in the middle, and then a tiny 4th ventricle just near the brain stem. This cerebrospinal fluid is made within these ventricles, circulates through them, circulates down that central canal of the spinal cord, and then circulates around that subarachnoid space that I pointed out, around all of the neural tissue. Here you can see that path of circulation. As I mentioned, that lateral ventricle, the 3rd ventricle, the 4th ventricle is here. These special places called choroid plexus is where that cerebrospinal fluid is made. It circulates through, travels down special features of our brain, goes through that central canal of our spinal cord, and then up through that subarachnoid space surrounding the entire central nervous system. At special locations called the arachnoid villi, and you see that over here, the CSF will be reabsorbed back into the blood, so this allows for the CSF to be cleaned. This flow chart over here is a very good flow chart for looking at the flow of CSF through the brain and spinal cord. We'll stop here for part 1.